In this video, I'm going to show how DNA is turned into proteins through the process of transcription and translation. DNA strands are coded instructions that control the production of proteins in the cell. This process is called protein synthesis. DNA holds the genetic code that is used to synthesize and make proteins. Different segments of this code are called genes. One molecule of DNA contains many different genes. Many proteins are enzymes, which catalyze and regulate chemical reactions. So a gene that codes for an enzyme to produce pigment can control the particular trait. Here we see the double-stranded DNA, and the phosphate bases are adenine, cytosine, guanine, and thymine. We can also see that RNA is single-stranded and has the same bases except for thymine, which gets replaced by uracil. There are three types of RNA. Messenger RNA, or mRNA, copies the DNA sequence. Ribosomal RNA, or rRNA, is in the ribosomes, and transfer RNA, or tRNA, transfers amino acids. So here we have the cell and nucleus. Protein synthesis occurs in the nucleus and cytoplasm of the cell. It occurs in two parts. Transcription, which is using the DNA to create a sequence of RNA, and translation, which is using the sequence of RNA to create a protein. Now we're going to talk about the process of transcription. The RNA polymerase comes in and unzips the DNA strand. The DNA is used as a template to make the mRNA. The RNA slides along the DNA and reads the base nucleotides from the DNA and places complementary bases on the RNA strand. C and G nucleotides match up, so does A and T. But anytime there's an A on the DNA, instead of laying down thymine, it lays down uracil on the RNA strand, thus creating mRNA. So how does the RNA know where to start? It has promoters, or specific sequences of DNA that say start. It also has a stop sequence, so the RNA polymerase knows where to end. After it reaches the stop, the mRNA is complete and then leaves the nucleus. This completes transcription. In short, this process of copying certain sections of DNA strands onto mRNA is called transcription. The mRNA travels outside the nucleus into the cytoplasm, where it will find a ribosome. Ribosomes can be floating in the cytoplasm or attached to the rough ER. Now let's move on to the process of translation. So translation happens three bases at a time, or one codon. A codon is a set of three adjacent nucleotides in mRNA. mRNA attaches to the ribosome, and the first start codon is read. The first amino acid is brought in by the tRNA. The anticodon on the tRNA matches up with the codon on the mRNA. Then another tRNA comes down next to it. The amino acids link together with a peptide bond to form a protein. Once the amino acids link together, the first tRNA detaches and makes place for the next tRNA to come in. This goes on, and the protein grows until the mRNA reads the stop codon. The codon acts as a code for specific proteins. Once the stop codon is reached, the new protein is made up of amino acids, is ready for folding. This completes translation and the end of protein synthesis. Thank you, and I hope you enjoyed this presentation.